Hello, everyone, and welcome to today's presentation. I'm excited to have you here as we explore the comprehensive overview of respiratory system anatomy and function. On this first slide, we see the title of our talk, along with the subtitle, From Conducting Airways to Alveolar Gas Exchange and Clinical Implications. Over the course of this session, we'll take a close look at how the respiratory system works, starting from the airways we breathe through all the way to the gas exchange happening in the alveoli, and what this means for clinical practice. So, let's get started. Let's take a closer look at the conducting airway anatomy. It all begins with the trachea, which is essentially the main airway that carries air from the upper respiratory tract down into the lungs. From there, the trachea splits into two main stem bronchi, one heading to the right lung and the other to the left. These main bronchi act as conduits, guiding air into the lungs without any gas exchange happening at this stage. Moving further, from each main stem bronchus, we see the emergence of lobar bronchi. These are the next level of branching, and they play a crucial role in directing air specifically to each lung lobe. Think of them as the highways that route air efficiently to different parts of the lungs. Then, these lobar bronchi divide even more into smaller branches called segmental and subsegmental bronchi. These smaller airways continue the process of conduction, getting the air deeper into the lung tissue. It's also important to understand the extensive nature of this branching system. The airways follow a dichotomous pattern, meaning each branch splits into two, and this process continues through about 23 to 27 generations. This creates a highly intricate network that ensures air can reach every corner of the lungs efficiently. All these structures work together seamlessly to facilitate effective air conduction, setting the stage for gas exchange to occur in the alveoli. This overview highlights just how complex and well-organized the conducting airway system really is. Let's take a closer look at the dead space volume in adults. On average, it's about 150 milliliters. This is the portion of each breath that fills the conducting airways but doesn't participate in gas exchange. It's important because it represents the volume of air that doesn't directly contribute to oxygenating the blood. Now moving on to the tidal volume, the amount of air inhaled or exhaled in a normal breath. In adults, this is typically around 500 milliliters. So, when you breathe normally, about 500 ml of air moves in or out with each breath. But here's an interesting point. A significant part of that breath, roughly 30%, actually fills the dead space. That means nearly a third of each breath is just filling the airways where no gas exchange happens. Understanding this helps us appreciate how efficient our respiratory system really is, and it sets the stage for discussing how various factors can influence gas exchange and overall breathing effectiveness. Now, let's take a closer look at the role of the terminal bronchioles. These tiny airways mark the end of the conducting zone, which is responsible for moving air into the lungs. Beyond this point, the bronchioles divide into respiratory bronchioles, where the crucial process of gas exchange actually begins. When we talk about the structure of bronchioles, it's important to note that they are very small, less than 1 millimeter in diameter, and their walls lack cartilage, unlike larger airways. Instead, their walls are made up of smooth muscle, which plays a vital role in controlling airflow. This smooth muscle can contract quite powerfully. During conditions like bronchospasm or bronchoconstriction, this contraction can actually cause the airway to collapse temporarily, making it harder for air to pass through. So, understanding the significance of this smooth muscle helps us see how the bronchioles regulate airflow and why conditions like asthma can cause such difficulty breathing, because the muscle tightens and narrows the airway. This connection between structure and function really highlights how finely tuned our respiratory system is, yet also how susceptible it can be to certain conditions. Let's delve into the role of the acinus in gas exchange. The acinus contains all the alveoli responsible for this critical process. It's essentially the site where oxygen from the air we breathe enters the blood, and carbon dioxide, a waste product, is removed. This exchange is vital for maintaining the body's overall oxygen supply and removing toxins. Now, looking at the structure of the acinus, it includes all the airways and alveoli beyond the terminal bronchioles. Think of it as the functional respiratory unit of the lung, where the actual work of breathing and gas exchange takes place. This organization allows the lungs to efficiently perform their role in respiration. Focusing on its significance, the acinus is the primary site for respiration in the lungs. Its proper functioning is crucial for maintaining effective lung performance and, ultimately, our overall health. Without a well-functioning acinus, the entire respiratory process could be compromised, highlighting its importance in our respiratory system. Let's take a closer look at the pores of Kohn. These are small openings that connect neighboring alveoli, and their main role is to facilitate collateral ventilation between these tiny air sacs. This means that if one alveolus is blocked or damaged, air can still reach it through these connections, which is crucial for maintaining proper airflow and efficient gas exchange. Essentially, they help ensure that even when some pathways are compromised, the lungs can still function effectively. Now, moving on to the canals of Lambert. These are channels that connect the terminal bronchioles directly to nearby alveoli. Their significance lies in providing alternative routes for airflow within the lungs. If the primary airways are obstructed, these canals allow air to bypass the blockage, helping to keep the alveoli ventilated. This mechanism is vital for preventing lung collapse and maintaining overall lung health, especially in cases of airway obstruction. Both the pores of Kohn and canals of Lambert are essential components of the lung's architecture. They work together to support continuous airflow and gas exchange, even under challenging conditions. Understanding these structures highlights how adaptable and resilient our respiratory system truly is, which ties into the overall theme of lung anatomy and function. Let's take a closer look at how the airway supply works within the lungs. Each segment of the lung receives air from its own segmental bronchus. This means that every segment has a dedicated airway pathway, which is crucial for efficient airflow and targeted treatment if needed. Moving on to the anatomical organization, the lungs are divided into lobes, and these lobes are further segmented. This regional division helps us understand how different parts of the lung function and how diseases may affect specific areas. Finally, it's important to recognize that each lung segment is supported by its own vascular supply. This unique blood supply not only sustains the health of each segment, but also allows for localized lung function. Overall, this detailed division and organization are fundamental to understanding how the respiratory system operates both in health and disease. Now, let's focus on some clinical aspects related to the respiratory system, specifically cystic fibrosis and atelectasis. When we look at a chest X-ray of a patient with cystic fibrosis, we often see areas of atelectasis, particularly in the right middle lobe. This happens because mucus tends to plug the airways, causing that part of the lung to collapse. It really underscores how vital imaging is in diagnosing and understanding complications that come with CF. Moving on to the role of respiratory therapists, they play a crucial part in managing these issues. For example, they might use gravitational drainage techniques to help mobilize and clear mucus from the lungs. These methods are essential because they help prevent mucus buildup from causing further lung problems. Gravitational drainage takes advantage of patient positioning. By carefully adjusting how the patient is seated or lying down, we can encourage mucus to move out of the lungs more effectively. Proper positioning can make a big difference in how well airway clearance works, especially in patients with cystic fibrosis who often struggle with mucus retention. So, in summary, recognizing atelectasis through imaging and utilizing targeted techniques like gravitational drainage are key components in managing CF-related lung issues and improving patient outcomes. Now, let's take a closer look at airflow velocity and how it changes as air moves through the respiratory system. As gas approaches the alveoli, the airflow velocity decreases sharply. This might seem counterintuitive at first, but it makes sense when you consider the large increase in total airway cross-sectional area that occurs due to dichotomous branching. Essentially, the airways split repeatedly, spreading out the airflow and slowing it down as it reaches the tiny alveoli where gas exchange happens. Moving beyond the terminal bronchioles, the way gas moves shifts significantly. Instead of bulk flow, it primarily occurs through molecular diffusion. This diffusion process is vital because it allows gases like oxygen and carbon dioxide to move across very small distances efficiently, especially in the smaller airways where flow velocity would be too slow or insufficient for bulk movement. Now, let's talk about airway resistance. Interestingly, airways less than 2 millimeters in diameter contribute only about 10% of the total airway resistance. This tells us that the majority of resistance is actually found in the larger airways. Also, it's important to note that the upper airways exert more resistance than the lower airways. This distribution of resistance plays a crucial role in airflow regulation and can impact breathing efficiency, especially in conditions where airway narrowing occurs. All these points tie back to the overall theme of how our respiratory system is finely tuned to optimize gas exchange while managing airflow resistance efficiently across different regions. Now, let's take a closer look at the composition of the lamina propria. Beneath the respiratory epithelium, this layer is made up of several important components. We have smooth muscle, which provides flexibility and support, elastic fibers that help the tissue withstand stretching, blood vessels that supply oxygen and nutrients, and nerves that are essential for regulating airway functions. Together, these elements work in harmony to maintain the structural integrity of the airway and support its proper function. Moving on, let's discuss the structure of the conducting airway lumen. This lumen is lined by what we call pseudostratified columnar epithelium, often referred to as respiratory epithelium. This specialized lining is crucial because it not only facilitates the passage of air, but also plays a vital role in protecting the airway from pathogens and debris. Its structure is tailored to support both airway function and defense mechanisms. Now, when we look at the formation of the respiratory mucosa, we see that it results from the combination of the epithelium and the underlying lamina propria. This mucosa forms a critical barrier and functional layer that helps in clearing particles, trapping pathogens, and supporting overall respiratory health. It's a key component in ensuring that our airways remain open, protected, and capable of efficient gas exchange. Now, let's talk about the crucial role of cilia in our respiratory system. Each ciliated epithelial cell typically has around 250 tiny hair-like structures called cilia. These cilia beat very rapidly, approximately 1,300 times per minute, in a coordinated fashion. This rapid beating is essential because it propels mucus upwards toward the pharynx, helping us clear our airways of debris and pathogens. Moving on to mucus secretion, goblet cells along with submucosal glands produce mucus that coats the epithelial surface. This mucus forms a protective blanket that traps harmful particles, like dust and microbes, preventing them from reaching the lower respiratory tract. Now, the histology of the conducting airways shows us how important this mucociliary system is. The coordinated movement of the cilia is vital. It ensures that inhaled particles and pathogens are effectively removed. This synchronization maintains a clean and functional airway, which is fundamental for healthy breathing and overall respiratory health. In essence, the cilia and mucus work together as a dynamic team to protect and keep our airways clear, supporting the broader function of the respiratory system. Let's take a closer look at how the respiratory system keeps itself clean through the ciliary movement mechanism. Cilia are tiny hair-like structures that extend into the gel layer of mucus. They perform a rapid, forward stroke that moves mucus toward the pharynx, helping to clear debris and pathogens from the airways. After this, there's a slower, backward recovery stroke through the sole layer, which resets the cilia for the next beat. This coordinated movement is essential for maintaining clear airways. Now, regarding mucus production, our bodies secrete about 100 milliliters of mucus daily. This amount can increase significantly in conditions like chronic bronchitis, cystic fibrosis, or asthma, where mucus buildup becomes problematic. The mucus blanket itself is made up of two layers: the gel layer, which traps particles, and the sole layer, which allows the cilia to move the mucus efficiently. This entire process is part of what we call the mucociliary clearance system. It's a vital defense mechanism, but it can be impaired by factors such as cigarette smoke, air pollution, or dehydration. These factors diminish the efficiency of airway clearance, making it harder for the respiratory system to protect itself and stay healthy. Understanding these mechanisms highlights the importance of maintaining good airway health and minimizing exposure to these harmful factors. Let's now look at the impact of endotracheal tubes on humidity. When an endotracheal tube is used, it effectively shifts the isothermic saturation boundary, or ISB, deeper into the lower airway. This shift can lead to a humidity deficit, which is significant because it impairs mucociliary clearance and compromises airway defenses. This highlights just how important it is to maintain proper humidity levels during airway management. Now, moving to the role of the upper airways in humidity regulation. The nose, pharynx, and trachea work together to warm and humidify the inspired air, which initially enters at room temperature, around 22 degrees Celsius with 50% relative humidity. These structures modify the inspired air, bringing it to body conditions, 37 degrees Celsius and 100% relative humidity. This process occurs at the isothermic saturation boundary, or ISB, which is essentially the point where the air reaches full saturation and is properly conditioned for optimal respiratory function. Understanding this process underscores why maintaining adequate humidity is so vital for respiratory health and effective gas exchange. It's a key part of ensuring our airways can do their job efficiently, especially when medical interventions like endotracheal tubes are involved. Now, let's take a closer look at some of the cellular players involved in respiratory inflammation. First, eosinophils are a type of inflammatory cell that infiltrate the airways during lung inflammation, especially in cases of allergen-triggered asthma. These cells are closely linked to allergic responses, and their presence is a hallmark of allergic airway inflammation. Next, we have neutrophils, which respond primarily to bacterial infections. They act by phagocytizing or engulfing pathogens, playing a critical role in fighting off bacterial invaders in the airways. Their action is essential in preventing bacterial infections from worsening or spreading. Moving on, we also need to consider the role of non-epithelial cells in the airway, especially mast cells. When activated, mast cells release inflammatory mediators such as histamine, prostaglandins, and leukotrienes. These substances cause mucosal swelling and smooth muscle contraction, which contribute directly to airway narrowing. Understanding these cellular mechanisms helps us see how inflammation leads to airway obstruction and symptoms like wheezing and shortness of breath. It's a complex interplay, but recognizing these key players highlights potential targets for therapy and management of respiratory conditions. Now, let's take a closer look at the vast gas exchange surface of the respiratory system. The adult lung contains approximately 300 million alveoli, which collectively provide an expansive surface area of about 50 to 100 square meters. That's roughly the size of a tennis court. This huge surface area is absolutely essential because it allows for efficient exchange of gases, oxygen in and carbon dioxide out, during breathing. Moving on to the alveoli themselves, they are quite small, measuring between 100 to 300 microns in diameter. But their small size is actually a key feature, as it's optimized for close contact with the surrounding pulmonary capillaries. This close proximity ensures that gases can diffuse rapidly and efficiently between the alveoli and blood. Speaking of capillaries, they are just large enough to allow red blood cells to pass through in single file. This tight fit is no accident. It maximizes the surface contact between blood and alveolar air, facilitating rapid and effective exchange of oxygen and carbon dioxide. So, in essence, the combination of millions of tiny alveoli and the finely tuned capillaries creates a highly efficient system for gas exchange, which is at the core of our respiratory function. This intricate anatomy underscores just how well designed this system is to meet our body's oxygen demands. Now, let's talk about the impact of diseases on the respiratory system. Certain diseases can actually increase the permeability of the alveolar capillary membrane. This means that the barrier, which is normally quite selective, becomes more leaky. When this happens, it can lead to pulmonary edema, which is essentially fluid accumulating in the lungs. This fluid buildup makes it much harder for gases like oxygen and carbon dioxide to be exchanged effectively. Moving on to the structure and function of this critical barrier. The blood-air barrier, or alveolar capillary membrane, is incredibly thin, less than half a micron thick. This ultra-thin design is essential because it allows gases to diffuse rapidly between the alveoli and the blood. At the same time, it normally prevents liquids from passing through, which helps maintain efficient and clean gas exchange. So, understanding the structure and how diseases can alter its permeability really highlights the importance of preserving the integrity of this barrier to keep our respiratory system functioning properly. Now, let's take a closer look at the types of cells that make up the alveolar epithelium. The first are the type I alveolar cells, which are flat and cover most of the surface area of the alveoli. Their thin, expansive shape is essential because it allows for efficient gas exchange between the air in the alveoli and the blood in the capillaries. Then we have the type II alveolar cells, which are more compact. These cells play a crucial role because they secrete surfactant, a substance that reduces surface tension within the alveoli. Surfactant is vital for keeping the alveoli stable and preventing them from collapsing, especially during exhalation. Moving on to alveolar macrophages, these are the immune cells that patrol the distal air spaces. Their primary job is to engulf and digest microorganisms, dust, and other foreign materials that reach the alveoli. They act as a first line of defense, helping to keep the lungs clear of potential pathogens and debris. Beyond their role in clearance, alveolar macrophages can migrate to other parts of the lung, such as the airways, interstitial spaces, or even the lymphatics. Some remain in the alveoli after they've ingested debris, continuing their surveillance role. This migration and adaptability are key to maintaining overall lung health and ensuring the respiratory system functions efficiently. So, in summary, the specialized cells in the alveoli work together. Type I cells for gas exchange, type II cells for surfactant production, and macrophages for immune defense, to keep our lungs healthy and functioning optimally.